Hello. Hi. Can you hear me? Yeah. Good afternoon. Perfect. Let's get started. Maybe some other people will join in. Okay. So we'll discuss previous year's week 10 questions. Jumping into the first question reads the process of weakening a pathogen while generating a vaccine is called what? Is it attenuation, vaccination, immunization, virulence reduction, or pasteurization? Do you know what happens to be the right answer? You can type it out from the chart or switch on your mic and say it.
Attenuation.
Yeah,
Attenuation.
yeah, attenuation.
Art.
Correct. Yeah, so it's called attenuation because you're weakening the pathogen. Okay, moving on. So, in immune isolated cell therapy, the cell should be within how much distance from the nearest blood vessel to facilitate effective diffusion of nutrients. So, immune isolated therapy is where you have non-host related cells, so coming from other person or other uh, animal, so on and so forth. But you're immune isolating them by basically surrounding them with this polymeric coatings or whatever the case may be. Diffusion permeate membranes, but how far away should the cells be from the nearest blood vessel to facilitate very effective diffusion of nutrients? Because these cells still require nutrients, right? So, what is the limit of distance you can place them from a blood vessel? Any idea?
I did not remember exactly, but it would be very close. So 500 micrometers.
Yeah, so yeah, you're correct. So 500 micrometers is the minimum distance within which diffusion can, or the maximum distance to which effective nutrient diffusion can occur. So closer the better, but 500 micrometers at least. Okay. Which of the following would antagonize immune isolated cell therapy? Pick two options. Cells that were grafted less than 200 to 500 micrometers from the vasculature, meaning blood vessels. Use of biodegradable polymer to encapsulate the cells. Use of mechanically stable non-erodible polymer to encapsulate the cells. Use of biocompatible polymer that does not allow fibrosis on the implant to encapsulate cells. Use of polymer that can filter out antibodies and T cells to encapsulate the cells. Use of a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells. Use of allogenic or autologous cells for encapsulating an implant, or use of polymer that allows free passage of small proteins but not cells and large complexes to encapsulate therapeutic cells. Which of the following would antagonize immune isolated cell therapy or work against immune isolated cell therapy? So definitely not the first option because we've discussed 500 micrometers is the maximum distance you can allow these cells to stay from vasculature. So cells grafted within the distance, so between 200 to 500 micrometers or less from the vasculature, they will help in immune isolated cell therapy. But what doesn't help? Is it the use of biodegradable polymer to encapsulate the cells? Use of mechanically stable non-erodible polymer to encapsulate the cells? Biocompatible polymer that does not allow fibrosis on the implant to encapsulate the cells, polymer that can filter out antibodies and T cells to encapsulate the cells.
Sir, I have doubt on this question, so I was not clear about
Yeah, what was the doubt?
like how how immune isolated cell therapy is different from the other other techniques.
Yeah, so immune isolated cell therapy is basically so if you are having some sort of loss of function. So let's say a person's pancreatic cells are not producing insulin as you want them. So The other options are basically to keep injecting insulin, or basically to do stem cell therapy and replace the pancreas, or doing this, right, immune isolated cell therapy. So in this, what you do is you basically take these pancreatic cells from another source, like a different person, animal, stem cells, even so on and so forth. And the problem with directly putting these cells into the body is your body's immune system will recognize these cells as foreign, and it will start attacking and killing these cells. So to prevent that from happening, what you do is you put a coating on top of the cell, like you encapsulate the cells. So you surround the cells with a polymeric coating or some sort of coating, which will allow nutrients to pass through the membrane, but it does not allow antibodies or immune cells to reach the cells. So they cannot have any effect on the cells. Does that make it clear? In that way, it's different. So you're isolating these cells that you're putting into the body, because if you don't isolate, there will be severe immune reaction against them.
Sir, is that like we're using immune cells, our own immune cells, as a vector or as in or delivery methods to deliver, like it
Yeah.
is similar to gene therapy or
No, 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 no. So it's different from T cell therapy and CAR T cell therapy and all those. So those, what you're doing is, so if a patient comes into the clinic and you want to do immunotherapy, what you will do is you will isolate these immune cells from the patient's bone marrow itself, and you take them out and you do whatever you want with them, and you put them back into the body. So in that sense, those cells are coming from the patient itself, right? So the patient's immune system will not recognize that as foreign cells. They will still be patient cells, but in this case you're not using patient cells, you're using foreign cells. So like I was saying, so a person comes in with this pancreatic failure. So you want to supply the patient with islets of Langerhans, the pancreatic cells, right? So what you can do is you can get pancreas, these islets of Langerhans cells from a different person, and you can put them in this patient. Now, if you put them directly, the patient's immune system will attack. But you isolate and put them, then it's fine. Yeah, so in that sense, it's different from CAR T therapy and other immunotherapies, because in immunotherapy, what you'll do is you'll get the immune cells from the patient itself. In this case, it need not necessarily, this is not immune therapy, this is immune isolated therapy. So basically, whatever you're putting in into the body of the patient, you're isolating it from the patient's immune system. You need not necessarily use immune cells. You can use any type of cell you want.
I got it, sir.
Yeah. Yeah. So in that sense, now which of these would cause negative effect towards the immune isolated cell therapy? Is it cells that were grafted within 200 to 500 micrometers from the vasculature? Probably not, because 500 micrometers will facilitate a very nice diffusion of the nutrients. Is it use of biodegradable polymer to encapsulate the cells, mechanically stable non-erodible polymer to encapsulate the cells, biocompatible polymer that does not allow fibrosis on the implant to encapsulate the cells? So basically something to prevent fibrosis from happening, or a polymer, using a polymer that can filter out antibodies and T cells to encapsulate these cells, or using a polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells, or use of allogenic or autologous cells for encapsulating an implant, use of polymer that allows free passage of small proteins but not cells and large complexes to encapsulate therapeutic cells. Any idea?
It may be use of biocompatible polymer that does not allow fibrosis on the implant to
Okay.
encapsulate
And
cells.
what could be the other option?
I'm not sure about that, but
Yeah, so if you look at the options, this option gets kind of our attention, right? Because they're saying they will use a polymer that allows free exchange of proteins and cells to encapsulate these therapeutic cells. Now, what will happen if you do that? Antibodies are proteins, so they will go into the cells. T cells, B cells, macrophages, all of these are cells. So they can also go into the this layer and reach the therapeutic cells. Now, if they go in, your immune cells are going in, your antibodies are going in, they'll destroy these cells, right? So that will not help immune immune isolated cell therapy. The other option is, so you do not you want fibrosis to not happen, because if fibrosis happens, remember fibrosis is this very thick lining of debris, extracellular matrix, and cells. So what will happen is, as the fibrosis builds up, the amount of nutrition that can reach into the cells slows down. So you would want to use a biocompatible polymer that does not allow fibrosis. But what you don't want to do is use a biodegradable polymer. Because why? If you use a biodegradable polymer, over time, whatever is preventing immune cells from reaching these therapeutic cells, that's degrading. It's slowly dissolving away. So at one point, your immune cells will be able to reach these cells, right? So you don't want that to happen. Yeah. So that, and the other option is you cannot allow proteins and cells to reach the cell, reach the therapeutic cells. Is that clear?
Yes, sir.
Okay. Moving on. So next question reads: what are the major challenges of immune isolated cell therapy? The options read: generation of immune reaction, fibrosis of implant over an extended period, fragmentation of the implant, attack by CD8 positive T cells. These are cytotoxic T cells. Uh, or is it that this approach can be used in immune compromised individual, or is it that the wide variety of polymers are available to encapsulate cells? What are the major challenges?
Generation of immune reaction.
Okay. So generally, whenever you're designing this immune isolated cell therapy, you will make sure that these cells and antibodies are not able to reach the site of wherever your therapeutic cells are, right? So I would say generation of immune reaction and attack by CD8 positive T cells is less of a concern, because you're planning it in such a way that they're not able to even access it in the first place. Now, what could be a major concern is, like we were discussing previously, fibrosis of the implant. So if there is this thick layer of extracellular matrix and dead cell debris that's forming on these polymers that contain the cells, it will limit the amount of food and oxygen that goes into the cells, right? So that's a major concern. If it's kept for a long period of time, fibrosis can happen. That will prevent these cells, therapeutic cells within the polymer, to survive. And the other one is obviously the fragmentation of the implant, right? So fragmentation is basically breaking of the implant. Now, if this implant is put into the body, and if the person is doing some strenuous activity or so on and so forth, what can happen is the implant can break. And if it breaks, that will immediately allow access to all the immune cells. Like your cells will get lost, they get washed away. Immune cells can come in and attack and all those things. So I would say, more than generating an immune reaction, these two are a big concern, because immune reaction, anyways, you're designing the polymer in such a way that it's not letting the cells and antibodies to come in. But if there is fibrosis, the cells will die from starvation. And if there is fragmentation, you're breaking the implant, then that will also cause severe failure of the device, right? So those two would be the major concern. For some reason, I guess the previous year answer had said generation of immune reaction. Sure. Uh, then attack by CD8 positive T cells could also be a major concern. But I can check back on this and come back what the correct answer is in the next class. But usually fibrosis is a major concern, and fragmentation is also a major concern with these implants. Okay. Moving on, which of the following methods can be used for encapsulating cells in biomaterial? Two options are available: spray drying, ionic gelation, emulsion based methods, microfluidics, hot melt, or lithography. Any idea? How can you encapsulate these cells in the biomaterial? Like you have this biomaterial. Now, how are you going to put cells inside this biomaterial?
So the first is emulsion-based method,
Okay,
and um,
Ionic
and
gelation.
spray
ionic
drying.
gelation. Okay. Yeah, so emulsion based methods we do not tend to use because emulsion based methods are very stringent, right? So what is emulsion based method? So it's oil in water or water in oil emulsion based methods, and cells cannot generally go into oil. They might have to go into water, and even then, what will happen is so for you to achieve a good encapsulation, you will have to have a lot of water and less oil. So the turnover is going to be very less. So ionic gelation is something that you can use, but there's also one more that's very popularly used. So it's definitely not hot melt or lithography because both of those will kill the cells.
Spray drying.
No, not really, because spray drying involves very high speeds and generating vacuum and basically use of current as well. So electricity to achieve these spray drying threads and put the cells there. So that will also kill the cells. So microfluidics is another option that you can use to sort of encapsulate these cells. So microfluidics is basically uh, let's say it's more or less like emulsion based method but on a much micro scale. So what happens is you have two taps. From one tap cells are coming out, from the other tap your polymer is coming out, and you release them both at the same time and they both mix and your cells get into the polymer. Essentially that. So you're letting your cells flow into the polymer. Okay. The next question reads: which of the following is a type of therapeutic vaccine? Two options are correct here. MMR
Cancer,
Sorry, Divya.
Cancer vaccine.
Yeah,
cancer vaccine.
yeah. Cancer vaccine is one of the therapeutic vaccines. What is a therapeutic vaccine? Something that you give before or after?
After the
Yeah. Okay. Yeah. Yeah. So cancer vaccine is one of the therapeutic vaccines. What is the other therapeutic vaccine? There's one more here. MMR, rabies, smallpox, polio, HCV.
I think rabies vaccine.
Yeah, so rabies vaccine is also a therapeutic vaccine. Uh, you can take the vaccine after you've been bitten by the dog and after you've been infected with rabies. Okay, moving on. Which branch of immunity is triggered when a vaccine is administered to an individual? Is it the humoral immunity, adaptive immunity, passive immunity, synthetic immunity, or random immunity? Yeah,
adaptive immunity and humoral immunity.
yeah, adaptive immunity and
Humoral immunity.
correct. Yeah, so what is humoral immunity?
Humoral basically means blood, so antibody mediated immunity. So
Correct. And what is adaptive immunity?
adaptive immunity is like there are two types of immunity, like natural and adaptive. Adaptive is a specific type of immunity that
Yeah.
is triggered due to the specific type of antigen, and it is um, what-
Yeah, you're correct. So over a period of time, it will evolve and develop an immunity. Uh, but how is humoral immunity different from adaptive immunity? Any idea?
so adaptive immunity also includes cells, different types of immune cells, and different reactions. And
Hmm.
humoral is only like antibody mediated
Correct.
reactions, immunizations.
Correct. Yeah. Any example that you can give for what type of cells are involved in adaptive immunity?
Sir, I have to read again.
Yeah, so CD8 positive cytotoxic T cells, right? So those those are part of the adaptive immunity. So T cells can generally have two types of phenotypes. One is CD4. There is third and fourth type as well, but we'll not discuss those at the moment. But generally, they can be CD4 positive or CD8 positive. So if a T cell is CD4 positive, it's called the helper T cell. Now what that will do is it will go and activate the B cells and ask the B cells to become plasma cells and produce antibodies. On the other hand, the CD8 positive T cells, which are the cytotoxic T cells, they also get triggered once there is antigen presentation from the major histocompatibility complex in phagocytic cells like macrophages, dendritic cells, so on and so forth. But those cells, they cannot go and activate antibody production. They will start killing, recognizing, getting trained on recognizing the infected cells and killing the infected cells. So adaptive immunity contains both humoral immunity as well as cell mediated immunity, uh, in the sense that humoral immunity is part of adaptive immunity. Yeah, but you're correct. Whenever you're administering a vaccine, both those get triggered. So humoral immunity where antibodies are produced, as well as adaptive immunity where cytotoxic T cells and immune cell memory and all those things are generated. Okay. Moving on, what is an adjuvant in the context of an immune response? Is it a molecule that inhibits proliferation? A
A
molecule that,
molecule that stimulates an immune response to target antigen.
yeah, so it's a molecule
Stimulus.
that stimulates an immune response to target antigen. Yeah, that's correct. So you have an antigen, you can increase your body's reaction to the antigen with an adjuvant. Can you guys name some commonly used adjuvants? There is one
Sir,
adjuvant.
I don't,
Sorry.
no, sir, I had not remembered names.
Okay, so alum is one.
Complete. Uh, there are two types. Like I remember, complete and incomplete. Uh,
Your voice is getting cut, I think. But yeah, so there are several adjuvants. Alum is one of the most routinely used adjuvant in clinics. Okay, moving on. Which of the following are undesirable properties of a polymer used for immune isolated implant? Uh, Undesirable property. So, which property should the polymer not
Toxic.
have? Sorry?
Toxic.
Yeah, toxic is one. It cannot be toxic. What should
Non-erodible.
you're saying it should not be non-erodible, so it should be erodible.
Yes, sir.
Okay, so yeah, so the problem with the polymer being an erodible polymer is the reason you're doing immunoisolated therapy is to basically keep the immune system of the patient away from whatever cells you're giving the patient, right? So if the polymer is erodible, it's going to go away after some time. So then your patient's immune cells can come and attack these therapeutic cells, right? So you want the polymer to be non-erodible. You don't want the polymer to be erodible. So toxic is definitely one, but what is the other? You want the polymer to be non-erodible to prevent the polymer from going away after some time. You want the polymer to be mechanically stable because otherwise, if it's not mechanically stable, any movement from the patient can break the implant. You want the polymer to be biocompatible, right? Because polymer is what is being exposed to the body. You want it to be well tolerated by the body. You don't want the polymer to be toxic, but you also don't want the polymer to be biodegradable because if it's biodegradable, that means it's going to dissolve in some time. Now, if it dissolves in some time, you're putting these cells from a different source into the patient, and all those cells are now outside in the body, and the patient's immune system can attack it, kill it, whatever it is, right? So, you don't want the polymer to be toxic, and you don't want the polymer to be biodegradable. Is that clear?
Yes, sir.
Yeah. Okay. Okay. Moving on. Which of the following is a potential disadvantage of using particle-based approaches while delivering vaccine to antigen-presenting cells? So, disadvantage of particle-based approach while delivering vaccine to antigen-presenting cells. APCs
The
take up
formulation can be unstable.
the formulation can be unstable. Okay.
And second is the APC takes particles in large quantities that might cause the cell to burst.
Okay. Okay, so yeah, so generally these antigen presenting cells, whenever they're taking up these particles, right, they do phagocytosis. So phagocytosis is limited by several pathways within the cell itself. So they do not necessarily take up large particles that will cause them to die themselves. They'll take up only as much as they can. You're correct in saying, or yeah, so you said the formulation can be unstable, right? Yeah, so the other thing is, so whenever you're designing these particle based approaches, right, so you can fine tune the stability of the particle. So you can know for how long the particle is going to be inside the body, at what rate it's degrading, so on and so forth. So that's not really a major concern. But the major concern comes in the fact that the formulation can increase the in vivo half life of the vaccine. Now, what will happen? So, if you put these whatever antigens are there and into the body, what can happen is they can survive for a very, very, very long time, even after the immune response has been generated, which is not necessary. And the other one is the antigen might lose integrity in the process of encapsulation. So if you're using a particle based approach, like we've discussed, either oil in water or water in oil emulsion. Now, if you're doing oil in water or water in oil emulsions for proteins, peptides, DNA, or in fact things like now recently RNA vaccines, right? So RNA is very unstable because of RNases. So what can happen is they can lose their integrity during the process of encapsulation itself. Is it clear?
Yes, sir.
Yeah. Okay. I think that's all the questions that I have from last year. Do you guys have any other questions or doubts that you want to discuss?
So the last question,
Okay. Let me share my screen.
like why not the other options are not correct?
Yeah. Let me share my screen. Yeah. So the other option is, like I was saying, antigen presenting cells, they know what the limit of phagocytosis they can undergo. So, for example, a macrophage, right? So it cannot simply keep on phagocytosing whatever is outside until it bursts. So there are pathways that prevent that from happening. So there is a limited amount of phagocytosis that a macrophage can do. So generally, they cannot take up particles in large quantities, and that can cause cell burst. That's not really a concern. And the other option is co-delivery of adjuvants is possible, which is true. Uh, so you can co-deliver. So basically, your antigen you're encapsulating in the particle, right? So at the same time, you can also encapsulate your adjuvant, whatever it is, into the particle. So that, yeah, so that in that sense, co-delivery of adjuvants is possible. In fact, a lot of people do it as well. All kinds of antigens, proteins, peptides, DNA can be loaded, which is true. So you can load anything you want as long as you're taking proper precautions not to degrade your antigen in whatever encapsulation conditions you're using. The formulation, yeah, this we've discussed. It allows local delivery of antigens. Yeah, so it does allow local delivery of antigens, which is an advantage. So here we are looking for disadvantages, right? So, for example, if you have an antigen encapsulated within the particle and you only want to deliver it to lungs, you can do that. You can put it in an inhaler and give the patient the particles through lungs. uh, Local delivery, very high local delivery. Similarly, you can deliver to different regions of the body by just injecting or doing an implant. So, local delivery is possible, and the formulation can be unstable. It's not true, because these particles that you're using, most likely PLGA microparticles and so on and so forth, they're very well characterized. We know the stability of them. We know how long they're going to remain as they are in the body, how when they're going to degrade, at what rate they're going to degrade, based on which molecular weight of the polymer that you've used. So higher the molecular weight, lower will be the degradation of the polymer, so on and so forth. So uh, not really a concern, and yeah, and it is also not a disadvantage, right? So yeah, so once you know at what rate the particle is degrading, you don't have to worry about, okay, whether my particle is going to survive in the body or not. You just have to worry about getting your like whatever antigen you have into the particle, which brings us to the final point, which is the antigen might lose integrity in the process of encapsulation. So, if you go back to your lectures, if you see how encapsulation can be done, you would have studied about single emulsion, double emulsion, right? So, what do you use for oil phase? Generally, it's DCM or organic solvents like those. Now, protein three-dimensional structure is very important for the proteins. If you use organic solvents like that, they'll destabilize the protein three-dimensional structure. Now, if that happens, the protein is useless. Even if the sequence is preserved, you're putting a protein that's basically structureless into your body, or wrong structure, so it will not elicit the proper immune responses. So, in that sense, losing the integrity during the process of encapsulation might be a disadvantage. So, you will have to take care of which organic solvent you're using, which aqueous phase you're using, so on and so forth.
Thank you, sir.
Sorry?
Thank you, sir.
Okay, okay. Yeah, any other questions you guys have?
So like there are two ways of delivery, mostly like viral and particular biopolymer based. So in what way we decide that in which mode of delivery we will go for
Yeah,
that one.
so trading, like if you look at historically, like if you want to deliver genetic material, right, like RNA or DNA, there was not a very good particle based platform to deliver them. So historically, what people have done is they have basically used viruses. Like the Covishield vaccine, I guess. One of the vaccines that was given in India, they used a virus, uh, some chimpanzee adenovirus. Now, what you can do is take this virus, remove all the disease causing genes, and put whatever gene you want into the virus, and that virus has you will not remove the infectivity of the virus, and you can put it in the body and it will start infecting wherever it is infecting, as it would normally do. But instead of causing a disease, it's going to deliver this vaccine or RNA or DNA sequence that you have in your mind into the body. Now, using viruses, you cannot deliver proteins or peptides. Uh, and of late, there are very good platforms to deliver DNA and RNA as well, using particles. Like the US like the Indian vaccine Covishield used a virus, but there's Moderna vaccine in the US that use particle-based RNA delivery, right? Liposome-mediated RNA delivery. So yeah, in that sense, the choice is mostly historical. Like what technique you're most comfortable with. Like, are you willing to pursue a new technique? Like, it all depends on the pharmaceutical company, right? Like if they're used to delivering RNA or DNA using viruses, they will not change their technique because why should they? They get their money using this technique. But yeah, so I hope that answers your question. So it's not really more of a choice. In fact, I would say viral based delivery poses certain risks that particle based delivery does not. So, for instance, viral based deliveries can still elicit immune response against the virus itself. So, which has happened against the adenoviruses. So, adenoviruses are the viruses that are routinely used to deliver this genetic material, antigens, right? RNA or DNA antigen. And what happens is your body can take up these virus particles and also develop immune response against the virus particle itself. So, the next time you're giving this vaccine, it will be less effective. But that's not necessarily the case with particles. You can fine tune the particles in such a way that they are not causing any immune response. The choice is mostly historic.
Okay, sir. Thank you, sir.
Yeah. Okay. If you guys don't have any other questions, then we can end the session here today. I'll see you guys next week. Okay. Bye.